First off, I'd like to welcome everybody to this presentation called Lab to Life, Understanding the Treatment Journey, and really sort of getting into the life cycle of research projects that ultimately make their way into clinical translation. So just a couple of points. So I'm Craig Walsh. I'm the chair of the Arthritis National Research Foundation Scientific Advisory Board. I'm also a scholar. I got my very first research grant when I first started as an assistant professor many years ago. I got a ANRF award, and so that really sort of helped to sort of set the pace for me. And I've been very excited to be involved with the ANRF since then. And I'm also honored to be your moderator today. So a big thank you to each of you and to our sponsor, Johnson & Johnson Innovative Medicine, for supporting today's discussion. And then, of course, I'd like to thank our panelists as well, Dr. Lenny Dragone, Nidhi Bhutani, and Eliza Tsou. And I'll give some more detail about those folks in a moment. So just to sort of set the stage for what we're going to be talking about today with our panelists, I just want to take a moment to set the stage and give you kind of a bigger sense of the picture that we're talking about. So when we think of new treatments, many of which are making the headlines, we often don't see what it takes years, sometimes decades of scientific research and toil in the laboratory that's behind all of those sorts of innovations. And so every therapy, it starts out as a bold idea, but it also has to go through a lot of different phases, including discovery, testing, validation, collaboration, and then eventually clinical trials and public access. And one thing that I'll say that's also really important to this is serendipity. Oftentimes, we have scientists, we have hypotheses about things, and we think, we design clever experiments to explain or to help us to prove those hypotheses. And oftentimes we find out that our hypothesis was wrong, and something far more interesting is really sort of there to explain what's actually happening. So we'll be sort of diving into that in our discussion today. So broadly speaking, the process essentially moves through 4 key stages. First off is basic discovery research, where there's a question or hypothesis that begins, and then we do more of that basic research to really understand, not just coming up with hypotheses, but also trying to understand mechanisms that are involved. And so a really important part of where we start is really understanding the basic biology of autoimmunity, rheumatologic disease, et cetera. And then there's translational and preclinical research where scientists take those early findings and begin to test and apply them in more targeted ways. There are clinical trials, which are also obviously very important and involve patients from the public. And those are really where these potential treatments are tested in people for safety and for effectiveness. And then finally, public access, when a therapy reaches patients and can become part of a standard of care. And of course, this isn't always a straight path. There frequently are roadblocks, and those kind of roadblocks include things like funding gaps, long timelines, regulatory hurdles, and in collaboration, as we'll see, is essential in pretty much every step in this process. So researchers, clinicians, biotech, and pharmaceutical partners all play a role. And then one thing I'll say about the Arthritis National Research Foundation that I think is essential, is part of this life cycle of therapy, it's sort of an interesting focus. Essentially, what we do at the ANRF is that we focus right at where the journey begins by funding the early stage, high potential research that can spark entirely new ways of understanding and treating autoimmune diseases. And in particular, one thing that I think distinguishes us from a lot of other funding organizations is that we invest in people rather than just in projects. And this, I think, is essential because it leads to dedicated researchers that are doing the important work of exploring the mechanisms, as well as the translation that's involved in coming up with therapies for autoimmune and rheumatologic diseases. So these are often the hardest projects that get funded, but they're also the most important for really changing what's possible in the future and giving us a new perspective that we can then use to take into therapies. So you're about to meet three ANRF supported researchers, each working at a different stage in this process. And together, during our panel discussion today, they'll take us from the light bulb moment to ultimately impact in patients, and to show how every treatment truly begins with a bold idea. So with that, we're going to talk about from idea to action, sort of the early discovery phase. And I'll be moderating this along with panelist Eliza Tsou. And so let's start talking about the very beginning when an idea is just forming. Dr. Tsou is an assistant professor of rheumatology at the University of Michigan, and her lab is investigating how epigenetics drives systemic sclerosis with the goal of improving our ability to predict and personalize treatment. And she was supported by ANRF from 2017 to 2019. So, Dr. Tsou, welcome.
Thank you. Thank you, Craig. I would like to first thank ANRF for the invitation. It is always fun to be part of a webinar that allows us to connect with patients and researchers in the field. For this first part, I would like to kind of walk everyone through how an idea becomes a viable research project and what is involved in the early stage research process. And so to walk you through that, I want to tell you a little bit about my journey and how I got here today. And so in my research, I focus mainly on systemic sclerosis, and this is a disease where there are three different components. There's tissue inflammation, there's blood vessel complications, and also tissue thickening in the skin and also in the internal organs. And so when I first started as a postdoctoral fellow, I was trying to think through I was hired as a fellow just to focus on systemic sclerosis, but I was trying to think what I want to focus on. So at the time, I decided that a main focus will be on blood vessels. I want to try to understand why patients, scleroderma patients, suffer from these vascular complications or symptoms that happen really early on in the disease. And at the time, I felt that this is something the field is missing. And so to answer that question, I started to think about what tools I need to use. As a young postdoctoral fellow, I thought that what I really need as a tool is these vascular cells from patients that allow us to really use and study them in experiments in a lab setting. And so for those of you who might know, systemic sclerosis patients have a lot of skin involvement, so we can routinely get skin biopsies from these patients. And from skin biopsies, we can isolate different cell types. When I was a postdoctoral fellow, most labs are focusing on fibroblast, fibrosis, skin thickening. So that's the one cell type people typically isolate. But for me to really focus on vasculature, I really need to isolate the vascular cells, which is very difficult to do. And I remember I spent my first year in my postdoctoral fellowship just to optimize to get these cells out from the skin to grow. And I did do that. And so with those valuable cells in hand, I was able to study them in the laboratory directly in different assays to study why they're behaving the way they are, why these patient cells are not behaving like cells from healthy controls. And we were able to really understand the behavior and how this behavior in a tissue culture setting can be translated into clinical features of the disease. And so fast forward from 2017 to now, leveraging with a lot of new technologies that we have to study these cells at a single cell level, we learned that these cells are potentially undergoing something we call cellular senescence, which means that these cells do not respond to biological signals and they don't behave normally, and they can trigger inflammation and change their behavior. And that's how these patients are suffering in their blood vessels. And so coming back to the overall goal of what is involved in the early stage research process, I would say it starts with identifying the research question, right? So for me, I decided to focus on the blood vessels of this disease. And so this process really involves a lot of reading and really getting to understand the disease that you're interested in, what has been done in the field and what the field has been missing. And once you set your stage, you have your hypothesis, and the next task will be how you design the study. Right. So what tools do you need to answer the question? And for me, I needed the tool from these vascular cells from the skin biopsy. That's the first thing that I did. And how I'm going to analyze the data. And then do I need collaborators to help me? So I'm a PhD scientist. I work in a clinical division, and with the biopsies that I need to isolate these cells, I do have to work with the clinicians and physicians in our clinical settings to help facilitate that process. So those are the collaborators I generated over the years. And then once we have all of those, then it's the process of research, right? So if you look at research, is re and search. So we repeating searching in the process. And this process can be long and it could be time consuming. And we spend a lot of time troubleshooting, rethinking our strategies. Oftentimes I tell my students, like, it's not rainbows and unicorns, right? When we have a hypothesis, you always have to spend a lot of time to kind of find that real answer. And so this whole process needs to be repeated until we get results that are reproducible and meaningful. And so to sum it up, it's a long journey in some ways. And for this, we do need a lot of time, effort, and resources to make it happen.
So on that last point, what role did the early funding from ANRF play in helping you to really sort of shape that, these early ideas that have become the basis for this?
Yeah, so I received support from ANRF back in 2017, and that was when I was a postdoctoral fellow trying to find my footing in the scleroderma field. And I'm very grateful that I received the funding from ANRF because that really helped me with the research that I was planning to do at the time, and also helped with my promotion into assistant professor and shortly after that. And so the funding from ANRF allowed me to have secure funding and also time to really dig deep into understanding the vascular cells that we were able to work with at the time. And in that funded project, we were able to determine the impact of a protein of interest and show that this protein plays a key role in both promoting the vascular complications and also fibrosis in the skin in two different cell types. And that really helped us understand how cells can communicate with each other in the skin. And also with the funding from ANRF, really early on, led us to further characterize the vascular cells that in more depth, and to lead us to the project that we are focusing on right now, which is cellular senescence. And I also want to add that funding from ANRF is not just the funding. I feel like it's more than just the financial support. It also offered mentorship and also networking opportunities within the scientific community, facilitating collaborations and knowledge, exchanging knowledge, such as the webinar today.
Yeah, exactly. And then I think one thing sort of that's key to this, like I said at the outset, that ANRF invests in people, right? Like yourself.
Exactly.
And, keep in mind that, in order for us to be able to do this kind of research, we need to get funds from other places. So oftentimes we're getting funds from NIH and other sources. And so, ANRF, I think, is really smart in the sense it's investing in people that they know are going to be successful, and they're going to be able to carry on that research for many, many years, as is often required to get this into the clinic. So, well, thank you for those answering those questions. That's great. So I'm going to move to the next stage, talking more about the sort of the translational and preclinical stage. And I'm going to be moderating this along with Nidhi Bhutani. And so the thinking here is really, once a research idea is underway, the goal becomes making discoveries that can help to expand our understanding of this disease. And so Dr. Nidhi Bhutani is an associate professor of orthopedic surgery at Stanford, where she leads a research on skeletal tissue regeneration and repair. She's an ANRF alum from 2014 to 2016, and a leader in cartilage and osteoarthritis research, exploring how stem cells and epigenetic changes can influence healing. So welcome, and thank you for joining us today, Nidhi.
Yeah, thank you so much for having me. This is such an important webinar to really sort of spread the knowledge of how difficult it is to really, do the early research and to actually carry it forward towards something which could be meaningful in patients. So let me tell you a little bit about my journey. As Craig mentioned, I was an ANRF fellow in 2014, And actually, we sort of started with the idea that epigenetics could be important for osteoarthritis and how to study that. And we were funded by ANRF early on to do that. And at that time, it was really an idea. We were looking at patient-derived cartilage from OA patients. And what we identified was that there were epigenetic changes which were very stark. So normally, we always think about epigenetics as something on top of the genetic material. So these are traits which are really affected by our everyday lifestyle, nutrition, a lot of things. And what we, with the help of ANRF funding, what we actually found was that epigenetics is actually a program which is very important in osteoarthritis. So this early research actually led us to believe that we can actually intervene in this epigenetic program to have a therapeutic benefit, which was a risky idea at that time, really. This is not an idea which I could have sort of run off to NIH with. And with the ANRF funding, and this is really critical for early stage recipients, that they're able to sort of explore these risky ideas, which can then turn out to be very, very useful. So now we have worked on this idea for more than a decade, and this is something which is really turning into a more translational idea, which we hope, we can take it further and maybe into patients at one point of time. So then what we did is, once you have this early discovery, right, we figured that epigenetic modification seems to be very important in osteoarthritis. Then we have to go back and look at mouse models, right, to try and get a mouse model of osteoarthritis and see what is the role that this modification is playing in this animal model. And it was very heartening to see that when we used a transgenic mouse, that we saw protection from osteoarthritis. But then to really make it into a drug, what you then need to consider is that now, is it a druggable target? Can you have a small molecule or RNA or antibody which can have access to this modification and hence would be useful in organism, so as to say, right? So that's the first step of a translational project. And then the second step is to consider whether this is something which is going to be useful in patients. And as you know, arthritis is a disease which is it is very heterogeneous. We have a lot of patients which are all thrown under this umbrella of arthritis, and it is really important to study the heterogeneity of these patients. So with the early funding, I was able to secure a R01 to first study this epigenetic modification in arthritis, and then actually a different R01 to sort of develop a platform to study these human tissues so that we can have some sort of an idea of whether this drug is going to work in patients. So as you know, arthritis is a disease for which there's a very high rate of drug failure. And that is because there's sort of a missing link between the animal models and the clinical trials. And that's kind of where this translational or the preclinical phase is so important. So what we have been able to do in my lab is to use really cutting edge techniques like single cell techniques to develop a platform where we use patient-derived cartilage tissues and really see the effect of a drug. So this is really important because, first of all, we can see clearly whether a candidate drug has a uniform effect. If it does, then that's good. It's like green light. It should go ahead. But we are also able to see drugs which fail. And importantly, using this platform, we are able to actually stratify patients, look at group A patients and group B patients, because it's very important to see maybe there's a drug which can work for group A, but it doesn't really work for group B. But then this is something where you can still take it forward and it would still be useful for this subset of patients. So this is this new idea of precision medicine, which is now coming in the translational phase that we are trying to develop in our lab. So in short, that is sort of like the journey in this phase to try and take a potential candidate drug forward.
So just kind of following up on that, I mean, one of the things, in my lab, we do a lot of work with genetically modified mice.
Mm-hmm.
And importantly, I think the viewer should understand that when we use genetically modified mice, one of the reasons that we do so is because obviously we can do experiments that we couldn't do in patient populations, but also because they're genetically identical mice, right? They're inbred over many, many generations. And so the real advantages is that we get rid of this sort of, polymorphisms and so forth, like all the genetic variation that exists in human populations. And so I'm wondering, when you think about stratifying these patient populations, do you think that's based on sort of the genetic background, or what are the sort of factors that contribute to one particular patient cohort being responsive to a particular therapy versus others?
So at this point of time, we are sort of trying to look a little bit closer or deeper into the tissue per se, because it is easy to look at one gene or one protein as an outcome and say whether the therapy is effective or not. So really, this tool is to first try and have a deep dive into the tissues that we are looking at. And what you're talking about is sort of like the next set, right? To be able to trace what are the comorbidities that these patients have, what is the genetic background of those patients, and those are also things which can then be, I guess, put into this platform. But the first thing is to really have robust readouts for the tissues that you are testing per se. So I guess our platform is really more geared towards doing that, having a deep dive into the outcome measures for these patients.
And just sort of kind of coming back to the question that I asked Dr. Tsou about, which is, tell us a little bit more about your journey with the ANRF and how ANRF was instrumental in helping you with your moving in your career. And tell us a little bit about what you're doing with the ANRF these days.
Yeah, so absolutely. So ANRF was really critical. The first grant that I ever received was from ANRF. And again, this was for, and I want to emphasize, it was a high risk, high reward idea, which is really important. And ANRF was sort of able to see the potential of this idea and fund it. And this is what sort of helped us move forward. I got funding from 2014 to 2016. And after that, in 2016, I was actually able to secure a major NIH grant, which really enabled me to follow this work further. And then we have been able to build up with that. And as Dr. Tsou said very nicely, ANRF is not just about funding, it's about really, a huge peer network, mentorship. And so that sort of really allows you to grow further. And recently, I've been very fortunate to be involved with ANRF in developing a peer group, alumni group, and leading that effort to really have more networking built in, where the researchers can, network with each other, get peer-to-peer mentoring, as well as have webinars like this, where we can sort of really take this out and do outreach and reach out and sort of explain why the scientific journey is so important and why this is an integral part of the community and how it really, gives back to the community in terms of moving medical breakthroughs forward.
Absolutely. I think of the investment of the ANRF, over the years, as we talked about before, an investment in careers and developing people who spend their lifetimes as doing research in these diseases, but also this idea that one of the things that I think is really value added for the ANRF's mission is this point about networking and providing mentorship to our folks. And I think it's a wonderful community that has developed. So definitely, as I said, sort of value added proposition.
Absolutely. I think it takes a village, and it's also important to know that it takes a long time. The timeline is like really huge, but then the benefits that you get out of them are also huge. So this is something which I think is important to remember. Absolutely.
Thank you, Dr. Bhutani. So we're going to move on to the next stage here. And we're going to talk about validation and collaboration. And I'm going to moderate this again with Dr. Tsou and with Dr. Bhutani. And so really what we're thinking here is, so let's say a discovery shows promise in the lab. What does it take to prove that it's real, that the results are real, that it's reproducible and ready to move forward in sort of clinical translation? So I'll start off with a couple of questions and then we can maybe extend that a little bit. So Dr. Tsou, what does it take to validate a discovery and show that it has real clinical potential?
Yeah, this is a great question. So validating a discovery and demonstrating its clinical potential is, in my mind, a very rigorous process that really involves a lot of critical steps. We need to first make sure that there is reproducibility and consistency in our data, right? And so this requires a lot of different experimental approaches that complement each other and really and ideally validated not by just one lab, but different labs. As us in academia and scientific setting, we always are trying to publish. And I do think that for reproducibility and consistency, we do have to go through, our data should go through that peer review process where publications are accessible to all the research labs that are interested in the disease. We also need to validate our findings in different models, tissue cell culture models, animal models that Dr. Bhutani has touched on. Now there are very popular 3D tissue models, bioengineered models, where I know that there's models that can mimic blood flow, similar to what we see in our blood flow in our body. So all of those validations we need to do not just in a one-dimensional model, but multidimensional. And then we really should dive into mechanistic understanding of the pathway and targets that we're interested in. And this findings there can help us anticipate potential side effects if we are developing a drug that targets a specific pathway that we're interested in. And then coming back to Dr. Bhutani's point on translational research, I think it is critical for us to kind of translate what we find in our lab and basic science and translate that into research and really determine how these findings that we see in our laboratory can be translated into therapeutic effects in more complex biological systems. Or clinical settings. And so this could include preclinical studies or clinical studies that can help us tease through that. And so again, this process, discovery and validation process, there is a long process, again, and requires a lot of scientific expertise, funding, time, and collaborative effort to make it happen.
So just kind of coming back a little bit to a number of folks that are watching may not know the life cycle of some of these research projects and the fact that, as you said, we have to get them peer reviewed, we have to get them published and get the work out there. So maybe you could talk a little bit about, that process and what are the sort of the challenges with that, but also why that's important for validation.
Right. So usually when we have a research project in mind, and then, the ANRF grant funds us 2 years, and NIH grants are 5 years. So it takes a lot of time to accumulate enough data for us to be able to publish something in a journal. And usually during this research process, we'll think about how we want to package our research idea into a publication, and that takes time. Writing takes time. Making the figures and look properly, it takes time. And then when we're ready to submit our findings into a journal, usually it will go through a review process. Sometimes it will take weeks, sometimes it's shorter. It depends on the journal that we submit the paper to, and then we will get the reviewer comments back, and it will give us some time to kind of work on our rebuttal, respond to the reviewer's concern. So that is a really rigorous review process, and then we can resubmit our paper for publication. So from project perception until writing until getting it published, it could take up to 2-3 years. If it's a really elaborate project. So it is a long process, but it is worthwhile because it goes through that rigorous review process that we all need to make sure that the data is reproducible.
And just again, for the folks that are joining us today may not know this, that process is anonymous generally. So you don't know who the folks are. Sometimes you can guess based on issues that they bring up, but and it's always reviewer 3 has the longest list of things
exactly.
that you have to do. But I think it's a really important point that people understand that science is generally self-regulating. I mean, and it is, we try to use best practices and we open ourselves to critique, and that process is not perfect, but it is anonymous and it does have some, I think, major advantages. So thank you for your comments with that. Dr. Bhutani, I think one key thing, as you're thinking about this concept of validation, but also collaboration as well, how do you measure success at this stage?
Yeah, so that's actually that is a $1 million question, right? Like, what do you think is now ready to move from the translation stage to clinic? I do think that having more of these platforms where we have patient-derived tissues or patient-derived organoids is important to sort of consider something which is our best guess for a drug candidate to then move forward to clinical trials. And this is also, in some sense, this is a place where you have a really requirement for huge funding, right, to go forward. So it's also is the step which sometimes we describe as the valley of death, right? Like to actually cross through from here to go to the clinical trial, you do need a lot of data showing robustness, showing reproducibility, which in some sense is self-regulatory and is a good thing. So I would say, yeah, once you sort of have all that data in animal models, in patient-derived tissues, that's what looks like success. Then this is also something where you really do need a lot of collaboration. So, for example, if I just talk about my own journey, whatever we do in one animal model, we actually collaborated with another group to have a different animal model. First we were doing a surgery-induced OA, and then we had ACL rupture-induced OA. And in having this collaboration, we were sort of much more, which was sure that our data was much more reproducible because we are doing it in multiple ways, in multiple sites, in different labs. So that's one thing. And then for setting up this huge platform of patient-derived tissues, of course, we have a lot of surgeon collaborators without whom it would not be possible. So you definitely do need collaboration at this stage. And then you actually need collaboration with biotech or, pharma to actually do a much more rigorous preclinical studies and then to take it forward towards clinical trials, of course. So I think, as I said before, it takes a village. So definitely collaborations are definitely key to making this translational phase a success.
Yeah, no, I absolutely agree. I mean, I think, we all have our own expertise in certain areas, but I think when, to kind of get to Dr. Tsou's point about how long it takes to get from, sort of early concept through all the, the lab, the research at the bench, and then ultimately getting it reviewed and accepted, it's a long period of time. And I think one of the things, especially as more technologies become available, those reviewers sort of expect that you're going to apply those technologies to answer some of these, questions. So I think one thing that you brought up that I thought was really interesting is this concept of, sort of evolving technologies, one of which being organoids. And it's important, I think, for the audience to understand that, we've had obviously animal models that we can, get really great, interesting data in, at the beginning, but we're not trying to cure arthritis in mice. We're trying to cure arthritis in people out there. So, ultimately we need to have analogs that, will sort of step in for the experiments that we can't do in patients. So think organoids, I just point out that we can take human stem cells and under certain conditions, those can be grown into models, for example, of joints and other kinds of tissues where we can do really direct experiments and ask really important questions with that. So I appreciate both of you guys. I thought that was a really great session. And then we're going to move on to the next, which is at the clinical stage, involving collaboration with industry, with biotech companies and big pharma. And again, I'm going to moderate this, and this is going to be with Dr. Lenny Dragone. And he's our final speaker. Dr. Dragone is a physician scientist, so he has both an MD and a PhD at the end of his name. So he's had training in both areas. And he began his journey in academic research and is now leading translational efforts in biotech, most recently as the chief medical officer at Abeta Biotherapeutics. He is an ANRF alum from 2009 to 2010 and still practices pediatric rheumatology at UCSF. So, Dr. Dragone, thank you very much for joining us. Just a couple of things. Maybe you can walk us through what it looks like to take a discovery sort of from the basic hypothesis, doing the bench research, and then ultimately moving that into a clinical trial.
Great. Yeah. Thank you, guys. Really wonderful foundation by Dr. Tsou and Dr. Bhutani. So everything that they spoke to is really foundational to identifying pathways, targets, and potential ways for treating patients with autoimmune and inflammatory diseases. I'm just going to spend a bit of time on the ANRF. They were also instrumental in my career early. That grant led to me getting an early funding from the NIH as well, which really led to me building mouse models of autoimmunity and inflammatory diseases in my own laboratory when I was in academics. And I am also still actively involved with ANRF. I'm actually involved with a venture fund that's being built out of the ANRF called Immune Boost Capital, and I am the active current treasurer of that venture effort from the foundation, with really the goals of that foundation taking some of the important discoveries made by Dr. Tsou and Bhutani and helping them bring them into early clinical trials. So really building a base of funding that will allow us to take their discoveries and others into that next step of clinical development. So, one of the most important things, and it was spoken about early, is understanding the right patient for the right treatment and finding the right match. So even though we call someone as having RA or having juvenile arthritis, those diseases are very heterogeneous. So there's lots of ways someone can manifest those clinical manifestations. So they look the same on the outside, but how they're getting there on the inside could be quite different. So understanding that biology, which is some of the translational work and the biomarker work that we talked about earlier, really helps us understand which are the right targets to prosecute to develop new medicines. So once that work is achieved, you can then identify new targets and new ways of targeting those pathways. And we develop things called new molecular entities, or NMEs, which is what we call them in industry. These NMEs can be all different kinds of modalities. They can be small molecules, kind of like things you take as pills. They could be biologic medicines that you could take either as an injection under the skin, or you can take them intravenously into your blood. They can also be novel therapies like lipid nanoparticles, which deliver specific molecules into cells, or even cells themselves. We can engineer cells and give them back to patients. So there's lots of different ways that we can develop new and novel therapies to treat different types of autoimmune and inflammatory diseases. In addition to understanding the biology and how to target that biology, one of the things we do when we're developing a new drug is really develop a comprehensive panel of studies done either in animal models, in preclinical animal models, in different species, as well as in even humanized models where we can take human immune systems, put them into animals, and actually test these molecules for safety. And that's the most important thing we do in early development before we dose a patient, is we go through a rigorous process of understanding the potential liabilities and benefits for the patient. And the liability piece is the most important part of what we learn early on, because that's all we can learn before we enter the official filings to the regulatory agencies. So to be able to develop a medicine in the world, there's different world regulatory agencies that review the totality of knowledge about those molecules. And in the US, that's the FDA that does that work. And we submit a very large document, usually thousands of pages long, called the IND, which allows us to then move these molecules into the first in human and first in patient studies. So that process is just building on all that basic knowledge that we learn, but then looking further at, is there risk at all for giving these medicines? All medicines have benefits and risks. So we're always trying to understand that, and obviously balancing those things when we bring new things into the clinic. So those filings are very important and comprehensive, and we work really closely with the FDA and the agencies around the world to ensure that what we are developing has a much greater potential benefit than any risks. Once that is done, then you bring those molecules into clinical development. That clinical development, depending on the molecule, can start sometimes in healthy volunteers. So what you end up doing in healthy volunteers is giving low doses and then escalating those doses up to understand the safety of those molecules in patients who are healthy, as well as understanding the biological effect. So as we start to develop molecules and dose them to patients, there are several things we look at. We look at the pharmacokinetics of those medicines. So when you give them to someone, how long do they last in the blood? How high do the levels get? And do those levels get to a point where we think they could actually be able to inhibit or influence the target at a successfully to actually have benefit for the patient? So all that work gets done initially in animals, but then has to be repeated in humans. So you understand that because some medicines, when you test them in animals, they perform a little differently. Their PK is different. So you have to understand what it's like in humans, because if you don't understand the pharmacokinetics of a drug, you may under or overdose it and give too little or too much. So those are important things to do early. We often do those in healthy volunteer patient studies. Some medicines only work in patients with the particular condition. So we are unable to do those studies in healthy volunteers, and we need to enroll patients with the underlying condition, whatever that may be, arthritis, so that we can actually look at the activity of those molecules in those patients. And when we do that, we work with a village, as everyone said here, it takes a village of folks to then, once we understand the pharmacokinetics of the drug, we then want to look at what's called the pharmacodynamic effects. So if you give the drug, do you see the biological effects in the target population of cells that you're trying to treat or the tissue? And you want to do that so you know that you're actually giving enough to get, again, that biological effect we think is going to lead to ultimately clinical benefit for the patient. And then in addition to looking at pharmacodynamics, we also look at biomarkers that help us understand, is the mechanism of action that we've developed in these preclinical models the same as it is in humans? So we do studies to look at the mechanism of action of these NMEs in patient blood samples. We usually look at circulating immune cells or at liquid biomarkers within the blood itself that tells us that, yes, when we give drug X, we see this biological effect, and that biological effect looks like we saw in those preclinical models. And then once we get to that point, we could start looking at biomarkers that could be surrogates for efficacy, for benefit for patients, and see if we're seeing changes in their immune cell function or in inflammatory markers in their blood that we think is going to ultimately lead to benefit for the patients. And then last but not least, when we design these trials, we design them to look at efficacy, to look at the benefits based on clinical measures. So we have, in many different autoimmune and inflammatory conditions, we have well-established measures of meaningful benefit for patients. So any of these medicines that we develop, we want to ensure that they actually have benefit that really exceeds what we have already out there for patients. Our goal is to develop new medicines that are really transformational and really offering patients new and greater advantages than what we can already offer them from FDA and from approved medicines. So we work towards that as well. So we kind of build that whole structure. We often also need to take a precision medicine approach when how we design these trials. And that gets at some of the information that both Dr. Tsou and Bhutani were saying. These diseases may only, and some of these pathways may only be present in a subset of patients. So as we're designing the trial, we actually utilize biomarkers to include specific subsets. So we have inclusion criteria for studies that allows us to enhance for the patient population we think is going to have the greatest potential benefit for the medicines that we're testing. Because we don't want to give people medicines we don't think will have therapeutic effect. We also don't want to expose them to medicines that have the potential for having safety problems. So we want to make sure we ensure that we're testing the medicines in the right patients as well. And then there's a long follow-up period afterwards, not only looking for benefits, for looking for any safety issues long-term in patients after we dose medicines. So that's kind of the broad strokes on how we then develop medicines and what kinds of measures we look for. Every condition has a different way of doing it, working with different physicians at academic medical centers who become the investigators on these trials. They actually will then recruit their patients into the studies, and then we will follow them together. We'll work together with them to follow patients over the course of the treatment and beyond the treatment as well.
Great, Dr. Dragone, that was a very thorough description of that process, and I definitely appreciate that. One question I have is collaboration between industry, biotech, big pharma, and academic researchers, and sort of how that process might work. Is that something that's simply at the sort of initial ideas and maybe taking that on, or is there an opportunity for some of that work that's happening in these academic labs to also impact biomarkers, for example, and really stratifying patient populations so that we can get drugs out there that are effective, at least for some folks?
Yeah, I think it happens at all stages, honestly. Some very initial discoveries that are done in academics really become the foundation for targets that you use for developing new molecular entities, these new enemies. Often you can have collaborations as you're designing the clinical trials. You want clinical experts from academic centers to be part of the construction of the clinical trial as you're going forward. They have unique insights into the diversity of the patients, into the clinical features of the patient's conditions that you would want to build into the clinical trial design as you're going forward. And then also in the interrogation of the samples that you isolate from the patients after the administration of the new medicine. So I think at all of those phases, there's really opportunity for industry and academic collaborations. And in all the trials I'm running, I have academic collaborations across all of those different parts of the process.
Great. Well, thank you, Lenny. I appreciate that. And I think we're going to take a few questions from our participants. So let me see here what we've got. Okay, so our first question comes from Sharifa Nurse. And the question is, can you tell me about platelet-rich plasma injections for pain? Does anybody want to take that one? That's a little outside of my pay grade, but
I mean, I can. So we have a collaborator who's been using PRP injections for OA patients, and what they see is what the whole field is seeing, that there is variability. There are some patients that benefit and some don't, and it is also not very well understood whether these effects are long term at this point. So what we are actually trying to do is sort of develop some blood-based biomarkers to try and analyze these patients and see what are the short-term effects, long-term effects, and whether we can actually have a more quantitative basis for assessing how effective these PRP patients would be for a particular patient. But that study, which is sort of just in early phases. I hope that answers your question. Yeah.
Great. Here's another question, and this is from Eliza. How do you know when a research idea is promising enough to pursue it further? And this is open to all of you.
Yeah, it's $1 million question. Well, it again comes to a lot of reading, really, and talking to your colleagues, talking to collaborators, come up with research questions that we all have an interest in, and we have the resources to do the research for the research idea, and do some preliminary studies to make sure that this research idea is feasible and realistic for the question that we're asking.
Great. Anybody else want to comment on that?
Yeah, I would say that, there's so many surprises in research. I think well-constructed, thoughtful research sometimes can surprise you. And I think just doing great science, and like the science that's been funded through the ANRF and other foundations, is really important, because sometimes you don't know when you start what's going to be really impactful for human disease until later. So I think just getting together a lot of really smart, good scientists who do good, careful science is really important, because sometimes you're surprised with what you think. Things will emerge from the research that really point you in a direction, and you didn't anticipate it when you first started, honestly.
Did you want to add anything to that, or should we move on to the next one?
Yeah, no, absolutely. I think it's the I totally agree that it's the unexpected results which actually sometimes, as a matter of fact, I would even go far and far on a limb and say that that's what actually causes the leaps in science. It's the unexpected discoveries which take us further than what we think is a incremental build up on what we already know. So I do think that this sort of like high risk, high reward research is very, very important, and we need more support for that. And I'm very grateful that ANRF is one of the foundations which supports this early ideas.
Absolutely. Okay, here's another question, and again, this is open to everybody. What would help researchers even more? Would bigger grants or longer research times, multi-years, help? Anybody want to answer that?
I do think that stability and just, if you can if there is a little bit more certainty that your ideas can be renewed for a longer time period, then that gives you the time and the funding required to actually build on those ideas. So, for example, sometimes the new ideas would have the research, but then to follow them for, a careful dosing study or something to actually make sure that this candidate that you have can develop into a drug, I think there is a little bit of a scarcity for those grants, and probably more foundations and more research support is needed there so that you can actually take it further, even in the academic circles, so that we can get it to the point where the baton can be passed on to biotech and for clinical trials. A little bit more support is needed on those fronts, I think.
And I'll add to that point, Dr. Bhutani, and that is you mentioned stability, and I think one of the things that's really essential, especially if kind of following up another point that was made about these unexpected results and serendipity in science. When we write research grants, for example, for NIH, we've got very, very specific aims that we spell out, and then of course we try to think about alternatives and so forth. But most of the time, when at least maybe 5 or 6 times in my career when we've had some like true breakthroughs, they've been totally unexpected results, and it's just been serendipity. So it's really hard to kind of anticipate that and put those into grants to some extent. You write around something interesting and just leave some space for that to happen. But anybody else want to comment on this particular issue? I mean, I would say that yes, both longer time and more money would be great for us to be able to do the work that we do. So that's a really great question. Here's a question for you, Lenny. What part of the research journey do patients often not get to see or hear about, but should?
Yeah, I'll focus on the stuff on the industry side. I think, how building really good new molecular entities takes a big village, right? Because depending on the target, the pathway, the biology, there's a lot of folks that are involved. If you're developing like a tablet, like a small molecule medicine, those medicines need to fit like a lock and a key into whatever the target is usually. And there's a lot of chemistry that gets done in this very elegant iterative way where they actually take the molecule you want to target, they crystallize it with these early compounds they find, and they do that over and over again. They kind of repeat that process to try to really make that lock and key most perfect. And the reason that that's important is because you want to make sure that these drugs are very specific to the target that you're developing it for, because you don't want it to bind to other things in the body that could lead to issues with safety. So again, it's thinking about how to be very mindful. And when you create some of these molecules, they can be metabolized by the liver in the body and they can become different. So really understanding really carefully when you develop molecules like this, how they can have an impact not only on the biology you want to modify, but on the body in general, takes really a group of very specialized scientists, toxicologists, clinical pharmacologists, and chemists, medicinal chemists, who work really in tandem together to really build. And that process can take years. It can take 8 to 10 years before you really successfully build the perfect molecule that gives you clean safety. So it's the same theme that you've heard from everyone else. This takes a long time, but it really takes a lot of very unique expertise as well.
Great. Okay, so we have one last question, and this is for all. What's one area of research that you're especially excited about right now? And I'll start with you, Dr. Tsou.
For me is the CAR T therapy that I think it's the new kid on the block this year. And I have been involved in some clinical trials here at Michigan doing CAR T therapy on patients with systemic sclerosis. And I think it is an exciting new therapy that it's quite promising, and we're very excited to see what the results would look like in a few years.
Dr. Bhutani?
So I think one major bottleneck for osteoarthritis has been that the disease is discovered very late in the process, so it becomes very difficult to actually reverse it. And we've been working on some blood-based biomarkers to try and capture the disease early. And this is something which I think could be a huge benefit to the patients, and that's something that we are very, very excited about. And
Dr. Dragone?
Yeah, so mine's a bit of a riff on Dr. Tsou's. I'm also excited about cell therapies, but what makes me really excited is the potential. They're very expensive and they're difficult to generate. So they have relatively limited a range of folks that can get them. I think what will really be the next advance is to be able to utilize ways to in vivo engineer cells to then allow them to get access and become like these CAR T cells that Dr. Tsou was talking about, where we can actually take that technology and bring it to scale around the world and really offer it to patients everywhere. But until we get there, there's some limitations on who will get access. And I think what's really important for all of us is not only to develop amazing medicines, but to develop medicines that allow access to every patient in the world who needs them. And that's a real, I think, a potential innovation that could enable that.
All right. I want to thank our wonderful panelists that have joined us today, and just wonderful conversation and great answers to some of those questions as well. And just to kind of close up about why early stage research matters, from this concept of this idea to ultimately treatment, the journey depends on bold questions, innovative minds, and critical early support from organizations, especially like the ANRF. And we're proud that over 98% of our scholars have remained in science, and many of them continue to contribute to the advancements that we've seen into these treatments. So thank you for joining us today and for being an important piece of the puzzle that keeps research moving forward. I also want to thank our partners at Johnson & Johnson Innovative Medicine for supporting this important conversation. And we hope that you'll continue this journey with us with our next webinar entitled Habit Stacking for Chronic Illness, which takes place on May 27th. And you can register for that and find all of the information on our events and resources for Arthritis and Lupus Awareness Month at our website, which is curearthritis.org. And with that, I'd like to thank everybody and look forward to seeing you on May 27th.
Thank you all.
Thank you
Take
all.
care, guys.
Thank you.